Hello, my name is Ali. Welcome to today's tutorial about 3D Flexible Refinement in CryoSPARC version 4.1 beta. 3D Flexible Refinement, or 3D Flex, has been developed over more than 2 years and was initially described in a preprint on bioRxiv in April 2021. To help understand the method and the details of using it, I would highly recommend reading the preprint. We've been working over the last while to improve the method and especially to make it more robust on a wider range of datasets without too much expert tuning of parameters. This work is still ongoing and we expect to make further modifications and improvements to the method, but it's now ready for CryoSPARC users to try out in v4.1 beta. An updated paper will also be coming out soon. So to get us started, I'll just give a bit of background about how 3D Flex works so we can use the right words and terminology to refer to various inputs and outputs when using it in CryoSPARC. 3D Flex is a deep generative model of continuous conformational heterogeneity in a single particle dataset. Like all generative models, it defines a forward process or a generative process by which a data point, in this case a single particle image, is generated under the model, and that model relies on some unknown parameters. And then there's a training procedure in which we try to estimate those unknown parameters from the data that we actually observe in the microscope. So in 3D Flex, the forward model involves, for a single particle image, first a latent coordinate. So there's one latent coordinate vector per single particle image in our dataset. That latent coordinate is unknown, but assume that we know it for the forward process. The latent coordinate vector is a vector from a latent space that has a certain dimensionality, and we can choose that dimensionality when we're setting up the model. That might be 1, 2, 3, 5, any smallish number for 3D Flex. The second part of the generative model is a canonical 3D density map. That's a density map that's supposed to represent the structure of our protein in its base conformation. So in one conformation from which all other continuous flexible versions of the protein are generated. So the generative process starts with the latent coordinates passing through what we call a flow generator network. This is a deep neural network that takes the latent coordinate and through a nonlinear process computes a deformation flow field that's denoted as U here in this diagram. The flow generator network has some parameters. Those are the weights of the neural network. Those are denoted by theta here. And the purpose of the flow generator network is to actually model the motion that's happening to the particle at latent coordinate z. So when latent coordinate z is 0, we're expecting that there isn't any deformation and our protein looks just like the canonical density map. As the latent coordinate moves around in the latent space, the flow generator network generates some deformations that, when applied to the canonical density using a convection operator, yield a convected 3D density map, and that's actually the density in a new position, in a new conformation under the deformation field that is present in the experimental data. That's what the forward model is simulating. So once that convected density map is constructed by literally deforming the canonical density map by using what the vectors that the deformation field generator outputs, that 3D density map, the convected density map, is then projected through the standard cryo-EM image formation model that involves projection by some pose and then corruption by CTF, added noise, and that yields what the model expects experimental images to look like. So again, that's the forward generative process of 3D Flex, and the idea of training 3D Flex is to use typical gradient-based backpropagation and gradient-based optimization to train both the canonical density that we don't know ahead of time and the flow generator network that we also don't know when we're starting, to optimize those to match the experimental data. In addition, because this is a generative model, we also don't know the latent coordinates of every particle. We don't know what conformation is present in each particle when we start, and that latent coordinate, essentially the position of the particle in the latent landscape or the conformational landscape, that also has to be estimated from the data during training time. And that's what we call autodecoder latent inference. So all of these three variables, latent coordinates, flow generator network parameters, and canonical density, are being jointly optimized during training against the experimental data. All right, so now that we know how the 3D Flex model works, we can see how it's implemented within CryoSPARC. In CryoSPARC, we've split up the 3D Flex workflow into 5 different job types to make it easy to iterate through the workflow and modify inputs and parameters along the way, and repeat certain parts depending on the kinds of outputs you're getting. Of course, like every algorithm within single particle data processing, especially more advanced algorithms, there's a lot of iterative work and a lot of investigation of results and visualization of results that's important as you're working with 3D Flex. So expect to run most of these jobs multiple times per dataset to see how to tune parameters and how to adjust things to get good results. 3D Flex is also in beta still within CryoSPARC, so we do expect some of this workflow, parameters, and etc. to change over time as we continue improving the method, and we may also consolidate some of these jobs or create new ones to make it easier to inspect results. So I'll just walk through the 5 different job types in order first, and then we'll dive into each one in an actual example. First of all, the 3D Flex data preparation job is responsible for taking your best set of particles so far and preparing those particles for use in 3D Flex training and high-res reconstruction jobs downstream. The data prep job does some basic pre-processing, whitening, etc., and stacking all the particles into continuous MRC stacks to make it easier for loading later on. And it also performs cropping and downsampling depending on the options that you choose. Now, it's very important that the particles that go into 3D Flex already be pre-processed as much as you can with the existing tools within CryoSPARC, and in particular because 3D Flex, as you'll read in the tutorial in our guide, does have a limitation that it currently is not built to deal with compositional heterogeneity. When there's compositional heterogeneity, density within the protein is appearing and disappearing. Subunits might be appearing or disappearing. There may be a binding partner or so on. And those kinds of changes are not well modeled as motion, as you can expect. So when there is compositional heterogeneity, the 3D Flex model can struggle and can provide results that are a bit strange. We'll discuss that later. So it's important that before you start, you perform the most cleanup that you can for your data, separate discrete conformational states, particularly those with compositional heterogeneity. It's okay to have discrete states that are still conformationally related versions of the same object. As long as there's the same densities present in all the different states, discrete heterogeneity is fine, and 3D Flex will be able to model that. But if you have compositional heterogeneity, try to separate that out beforehand. Once you have your best set of particles, put those all into a single refinement and try to get the best consensus or rigid refinement possible. And those final particles with their alignments and CTF parameters will be used in 3D Flex. And that's what's input to the 3D Flex data prep job. Once the data prep job is run, it also outputs a initial volume, an initial guess, essentially for 3D Flex to use, and that's simply equal to the consensus reconstruction or rigid reconstruction of all the particles. And it's appropriately cropped and downsampled to match the particle sizes. So the 3D Flex data prep job simply does all that, and you can basically run that once and you'll be able to reuse the same prepared data multiple times as you're changing the model and retraining. The next job is 3D Flex mesh preparation. We'll talk about meshes, tetrahedral meshes for 3D Flex in a moment. Those are quite important, and there are many different options to generate a mesh. By default, the 3D Flex mesh prep job automatically generates a reasonable tetrahedral mesh based on the consensus reconstruction, so the rigid reconstruction that you have right now before you run Flex Refinement. But you can also provide a segmentation file. You can provide other inputs to modify the mesh, or you can make your own custom mesh from scratch and input that into CryoSPARC using the new CryoSPARC tools system. And I'll describe all of that later as well. And in fact, I'll have a separate tutorial video about making a custom mesh using a segmentation in the 3D Flex mesh prep job. Once a mesh is defined from the 3D Flex mesh prep job, that mesh along with the prepared particles from the data preparation job go into a 3D Flex training job. And that's the main job that actually defines and then trains a 3D Flex model. So that job has parameters related to the architecture of the model, the number of neurons in the hidden layers, learning rates and other hyperparameters, as well as some very important hyperparameters to do with the rigidity prior that's part of 3D Flex. And for details of all these topics, please do read the preprint that describes all of this in detail. The rigidity penalty and other hyperparameters of the model do need to be adjusted on a per dataset basis. We've tried hard to sort of rework how these work to make it easier to find reasonable values, and we have some good defaults now that work in many cases, but we do recommend that users play with these parameters of the 3D Flex training job, and we'll describe those in more detail once we get to that job. Once the model is trained, and in fact during model training, which can take some substantial amounts of time, often several hours, up to 10 or 15 hours, depending on how large the data set is, although some smaller data sets can be trained in 3 to 4 hours. During training and after training, the FlexRefine training job outputs checkpoints, and each checkpoint contains a definition of the 3D Flex model, as well as all the model parameters. So those are, for example, the flow generator network parameters theta, the canonical density map v, all the latent coordinates, and all the hyperparameters that define the model. And so those checkpoints from the training job can be used for two things. The first is the 3D Flex generator. And the 3D Flex generator job is essentially a visualization tool that helps to create volume series, just like 3D variability display job, for those who are familiar with it, to create volume series that span across the latent distribution of the data set and help to visualize what the model is learning, what kinds of motions and conformational changes the model is learning. The 3D Flex generate job also has some other options that we'll talk about. It can perform the same generation operation on a high resolution input volume, so that you can also create a volume series from a high resolution density map that you've got from 3D Flex Reconstruct or other sources. And it can also perform generation with a custom trajectory through the latent space, and you can provide a custom trajectory using the new CryoSPARC tools system. So we'll talk about that job as well, but it's essentially used for visualization. And the nice thing about it is that it can run even if the training job is not finished. So as long as the training job has outputted at least one checkpoint, wherever it is in the training process, you can run a 3D Flex generate job and have it generate volume series, so you can inspect what the model is doing even while training is going on. The fifth job in the series is 3D Flex Reconstruction. And this is a job that's responsible for performing the BFGS-based high-resolution reconstruction that's described in the preprint. Essentially, this job takes high-resolution data particles from the 3D Flex data preparation job, as well as whatever checkpoint you choose from the Flex training job, and uses the 3D Flex model that's defined in that checkpoint to perform a high-resolution reconstruction of two separate half maps from the particle data. And those half maps are reconstructed under the 3D Flex deformation model. This means that for a flexible region of the protein, in every different particle, it appears in a different position. But the 3D Flex model is used to map backwards through the model and figure out how the signal from each particle can be put back together in a single 3D reconstruction at high resolution that contains signal from all the particles in the flexible regions. This is how we can actually improve the resolution of flexible regions under the FlexRefine model. What I'll do now is switch over to CryoSPARC and demonstrate these 5 different jobs on a simple data set. This is Empire 10345, an integrin particle that's known to have a lot of flexibility, and in particular a very flexible arm. What we'll do is run through the different job types from start to finish and explain the different parameters, choices, inputs, and the ways you can use these jobs to run the 3D Flex method. All right, so first of all, the most important thing to keep in mind before we start is something I already mentioned, which is that the particles that you put into 3D Flex data preparation should be the best particles you have so far. In this case, we have already purified these particles by separating out any conformational states using 3D classification, heterogeneous refinement, etc., whatever other tools you'd like. Any conformational states that contain compositional heterogeneity, i.e. there isn't the same density in every particle. And that's something, as I mentioned, that's not something that 3D Flex can deal with well yet. So what we've got here is a non-uniform refinement, job number 10, and this is with 84,000-ish particles. It gets to around 3.6 angstroms, and that's our best reconstruction so far. That's a consensus reconstruction or a rigid reconstruction, and the particles from that job, the output particles, which contain alignments 3D, as well as the blobs, which are the actual raw particle data, and CTF metadata, those particles are what get input into the Flex data prep job. Now, the Flex data prep job has several parameters. The first is a crop box size. This allows us to crop the input particles, which might be of quite a large size if we're just doing a standard refinement, And I'll just go back to this non-uniform refinement to see at the beginning of the log that our particles have a size of 300, so 300 box size, which is quite large, and for our current purposes is a little bit unnecessary for 3D Flex. One important note is that 3D Flex is a real space method, meaning it doesn't do Fourier back projection or alignment or any other comparison of particles in Fourier space. This means that it's possible to actually crop the box of particles a little bit tighter around the density and not have to worry about interpolation artifacts during Fourier interpolation. However, there is still a limit on how tightly you can crop the box around your particle because of CTF aliasing issues, especially if you have data collected at very high CTF and you're aiming for a very high resolution. In this case, that's not an issue, so we can go ahead and crop these particles a little bit. So in our data prep job, once again, the inputs crop box size we can set to 256, is what I've done here. The training box size can be set to 128. And this is an important distinction. So the crop box size, first of all, is applied directly to the input particles at their original pixel size. And that original pixel size will be used for for high resolution reconstruction in 3D Flex. Training happens at a different box size, a smaller box size. And those cropped particles are downsampled to this training box size. By default, we've set that to 128. You can change this value, but it's good to keep this as small as you can, while still maintaining a high enough resolution in the training particles to see the features that you want to use for modeling the motion. One important consideration, and this is something you can read more about in the preprint, is that for validation of reconstruction results from 3D Flex, it's important that the training box size be small enough that training is limited to a resolution, which will be the Nyquist resolution at this box size, limited to a resolution that's below the resolution you expect to achieve in your reconstruction. And that's because training of a 3D Flex model is not done with a gold standard split, since there's only one 3D Flex model and we have to train it jointly with all the particles together. And so there aren't two half sets of particles being used to train two different 3D Flex models. And therefore, all the particle data up to the resolution used at training time is available for the entire model to learn, and therefore we can only trust a resolution estimate that we get from half-set reconstruction or FSC comparison downstream if the high-resolution reconstruction that we've reached is beyond the training resolution that was used during training of 3D Flex. This is, again, described more fully and more clearly in the preprint. Generally speaking, a 128 box size is reasonable, and when you run this job, the job will print out information about the box sizes you've chosen and the resolutions that this will entail for training. So for example, input particles have a box size of 300 as expected. The input particle pixel size is 1.345 here. And we're cropping the input particles to a size of 256 and then downsampling those particles to a size of 128. And that means the Nyquist resolution at training time will be 5.4 angstroms. This is important because this means that during training, 3D Flex does not see any information about the protein from the data beyond 3.4 angstrom resolution. So that means if we find that after 3D Flex reconstruction, we're getting a resolution beyond 3.4 angstroms, we know that that extra FSC correlation and resolution that we're seeing is not due to overfitting in the 3D Flex model, since 3D Flex did not see any information beyond 5.4 angstroms. So that's a very important thing to keep in mind. The job then goes through and produces some diagnostic plots to show you what the consensus reconstruction looks like, downsampled at the training box size, so you can see how tightly the box is cropped around the particle, which is, again, not very tightly, but more tightly than the original particles were. The job goes through and processes particles in batches of 5,000 or whatever number is necessary to make the full set, and outputs those final particles. The other parameters of this job are minimum scale to keep. This is an advanced parameter that can limit particles or filter particles in the input based on their per particle scale factor. And so if you run a refinement that uses per particle scale optimization, you might find that there are some particles that have a very low per particle scale. So for example, in this non-uniform refinement, we can look at the scale factors that were estimated here, and we see that most particles have a scale factor around 1, and this per particle scale factor is essentially a proxy for the ice thickness of that particular particle. And estimating and correcting for the per particle scale, or using the per particle scale during back projection can often yield improvements in refinement, since particles with a very thick ice that will have a low per particle scale factor will end up being downweighted during back projection. But those particles may have a reason for having a low per particle scale factor other than just ice thickness. They may be broken, they may be denatured, they may actually be junk picks, or they may be misaligned. It's not a very clear-cut distinction, and it's not in all cases that the per particle scale factor actually separates junk particles from good particles, but in some cases it may be useful to just remove particles with a low per particle scale factor, just in case they do have some compositional heterogeneity or other issue that isn't good for 3D Flex. So we can do that by setting the min scale to keep parameter in our 3D data prep job. Finally, the last parameter is the number of particles to use. You can use this to limit the number of particles that will come out of this job. The number of particles has to be a multiple of 1,000 just to keep things simple in the 3D Flex training. And if you don't specify this parameter, the nearest 1,000 particles is used, in this case 84,000 particles, as the output for the job. So that's essentially everything about the data prep job. The particles that come out of the data prep job, if we look at the outputs, they have now a few different result types. One is blob_train, and these are prepared training data for this particle, this set of particles rather. They also have blob_full-res. These are the full resolution cropped particles, and they've also been pre-processed by whitening the noise and etc. The training blobs also come with CTFs. In order to keep the 3D Flex code simple, CTFs are pre-calculated. So the CTFs for both training and full resolution time are pre-calculated and outputted here as well. And then the original particle data, blobs, alignments, and CTF are passed through. Before we move on to the next job type, I'll just demonstrate how to create these 3D Flex jobs from the job builder. You'll find the 3D Flex jobs all in a section called Flexible Refinement, just after 3D Variability. And you see all 5 jobs there. You can also search for Flex Refine jobs in the search bar, and you'll find all the jobs there. The data preparation job, when you create the job, it requires 2 inputs, particle stacks and an initial volume. We'll get both of those from our best refinement job so far. So for example, the best refined volume from our non-uniform refinement, as well as all the particles. As I mentioned, these particles and volume can come from any source, but all the particles should be aligned together as best as you possibly can, since particle poses are not currently refined during 3D Flex optimization. Once you've connected those, this job is now built, number 153. You can set the set the parameters that you wish, and queue the job as usual. I won't do this now since the data prep is already done for this data set. I'll just get rid of this job to keep our tree clean. Now that our data is prepared, the next job in the set is 3D Flex mesh preparation. This is a job that is a little more involved and is responsible for creating the tetrahedral mesh that under which 3D Flex represents deformations of our protein molecule. When we create this job, I'll show it here. We can create a 3D Flex mesh prep job. It has 2 inputs. One is a consensus volume, and this should be the output from our 3D Flex data preparation job, which produced a consensus volume that's already downsampled and cropped to the correct training size that we're going to use, that we had set up previously. It can also optionally take a solvent mask, and you can produce this in any way you want, or you can use the solvent mask preparation parameters here in case you don't provide a solvent mask. This will allow the job to create a solvent mask for you. The solvent mask is quite an important piece, although it's not very important in terms of modeling flexibility. The solvent mask within 3D Flex simply defines the region of the canonical density that 3D Flex is actually going to optimize. Outside of the solvent mask, 3D Flex just assumes that the density is 0. And so it's important that the solvent mask be large enough to contain the flexible parts of the molecule that we expect to resolve. And just to be very clear here, the solvent mask is applied to is relevant to the canonical density, the canonical 3D map. So it defines which parts of the canonical 3D density map are allowed to have non-zero density. It doesn't apply to the convected 3D map. That means our deformation field can actually convect density from the canonical density map outside of the solvent mask, which is a good thing in case there are flexible parts of our protein that move long distances. We don't need to create a mask that contains all possible conformations, but just a mask that contains the density in the canonical frame of reference. So that solvent mask can be, like I said, produced by your own means and connected here, or using these parameters. The first one is filtering the input volume. So the input consensus volume that's input here will be filtered to 10 angstroms by default, and then thresholded. And this threshold is an absolute value, so it needs to be changed based on the data set that you're using and the density values present in it, as well as a dilation and soft padding distance. The final solvent mask, the final hard solvent mask that defines the boundary of where the canonical density can be non-zero, is the outer edge of this soft mask. And the soft mask is just used to softly transition between density and solvent. So these parameters can be set to create a solvent mask. It's ideal to make the solvent mask large enough to contain the density, but not too large, since the computational expense of 3D Flex training and reconstruction are primarily related to the number of voxels, or the volume, inside of the solvent mask. So it's a good idea to make it not too much bigger than is actually necessary. The remaining parameters of this job are related to the actual tetrahedral mesh generation. And in order to demonstrate what these parameters do, I'll just open up this job number 93, which has already run, and it's a flex mesh prep job. So in this job, I didn't change any of the default values for mesh generation. And what the job does is starts again with a consensus volume, low pass filters it to 10 angstroms, and from this plot you can read off the density values in the color bar here. So 0.5 was chosen to be a reasonable density where we can cut this 10 angstrom result to create our solvent mask. The solvent mask, of course, after thresholding is dilated and expanded so that it fills up all the holes and is continuous. And this mask, you can see, is now large enough to contain this very floppy part of the protein here. And the hard mask is the outer edge of that soft solvent mask. And finally, just as a sanity check, we get a plot of the masked version of the input consensus reconstruction, again confirming that we're not cutting into the density anywhere, but our mask is not a whole lot larger than it needs to be, and that's just about right. And then comes the generation of the tetrahedral mesh. So the parameter, firstly, that controls the size of the mesh elements is the base number of tetracells. And this parameter determines how many tetracells fit across the box. In other words, how large is each tetracell. So the base mesh that's used has a fixed number that you choose as a parameter of tetracells across the box, and it's just a regular tetrahedral mesh that covers the entire box volume. And by default, if you haven't provided any other parameters, the job will simply create that base mesh and then trim it so that we're only keeping the mesh elements that actually have any part of the solvent mask inside of them. So the mesh elements out in the solvent are not needed, and they're trimmed and removed. And these plots show a couple of things. The colored plots that show the actual tetrahedra are plotting out the tetrahedral index map, which is described in the tutorial in our guide, if you want more information there. And that simply shows for every voxel which tetrahedron is that voxel a part of. And so this plot ranges up to the number of tetrahedra. You can see here that our generated trimmed mesh, after culling the tetra that are not useful, has 491 points. That means 491 vertices in the mesh, and 1471 cells, or 1471 tetrahedra in the actual mesh. In this case, since we didn't provide any segmentation or other inputs, there's nothing in the submeshes or segmentation section. But after the basic mesh is generated, the next thing that happens is computation and generation of rigidity weights. Sorry, one extra note about submeshes and segmentation. This is an area where there's a lot more flexibility for user input and choice about the topology of the mesh. And there's going to be a separate tutorial video that will be available in our tutorial on the guide, covering how to use the segmentation tools and how to create an articulated mesh with a different topology than just the standard continuous mesh that covers the overall object. So moving on to rigidity weights. Rigidity weights are also described in the tutorial in the guide, but essentially rigidity weights are a single number for each tetra cell, or each tetrahedron. And the rigidity weights, some are generally ranging between 0 and 1, or they can be higher as well, but they're relative weights that determine on a relative basis how rigid do we expect our particle to be in every tetrahedral cell. And this is related to the rigidity penalty that's described in the preprint. So cells that are more rigid in the model, when the model is being trained, those cells will have a higher penalty if they're deformed by the model in a non-rigid way. Whereas cells that have a low rigidity weight will have less of a penalty for deforming them. And this can be quite a useful way to tune or help the method, guide the method towards deformations that we are interested in finding and not those that we're not interested in finding. So a typical example is that normally we expect that empty regions of our density, for example, in this in this integrin particle, empty regions of space around the protein, and especially in between domains that might move relative to each other, those empty space regions should be able to compress or expand as much as necessary to allow the neighboring regions of protein to actually move closer or further apart. We don't want to make empty space stiff, essentially, in the model. And so by default, the parameters are set so that the rigidity weight of the emptiest elements is 0.5, and the rigidity weights of the densest elements, those that contain the most density in the input consensus structure, those are 1. And that's the default. You can change the minimum rigidity weight. There's also a parameter that can stiffen low density regions, and that means it's kind of described in the help text here, but essentially regions that are not empty but contain low amounts of density at the periphery of the molecule are stiffened by an extra factor. And this helps with small molecules and other cases where maybe the data quality is less high, where you're finding that there may be overfitting or there might be strange motions around the periphery of the particle where some peripheral pieces are flying around a little too much and the model is spending a lot of its capacity modeling the motion of floppy peripheral elements of your protein rather than the actual heterogeneity that you're interested in modeling. And so you can stiffen up those regions and essentially cause the model to ignore density or be biased against modeling density in those peripheral areas. So there's a lot of flexibility here with the rigidity weighting, and this is also something that can be customized using the CryoSPARC tools system. There's an example CryoSPARC tools notebook in the CryoSPARC tools documentation that can show you how to customize rigidity weights if you are so inclined. All right, so I won't discuss the segmentation or rigidification of segments parameters for the mesh prep job, since that's covered in a separate video. But that essentially outlines what the mesh preparation job does. This doesn't take very long to run, and when you're creating it, again, you don't need to provide a solvent mask. The only constraint is that the solvent mask you provide or that you generate here should contain, as I said, all of the regions in the canonical density that where you expect to resolve structure or improve the structure that's already resolved. So you want to be careful that your solvent mask doesn't accidentally cut off flexible regions of the particle. For example, in this case with the integrin, this flexible arm has relatively low density and obviously poorly resolved density compared to the rest of the structure. And so we don't accidentally want to cut off this flexible arm in preparing the solvent mask. Now that we have this 3D Flex mesh prep job built, number 154, I'm going to just adjust its parameters and run this job on one of our nodes. This job does not require GPUs, but runs entirely on the CPU, and it's quite fast. As I mentioned, it'll run through the steps that we already talked about, generate our solvent mask, soft and hard, and then our tetrahedral mesh, and finish. so only a few seconds. One very nice thing about this job type is that it's possible to download the mesh in a PDB format to make it easy to visualize. And this PDB format is not actually used downstream within 3D Flex, since it doesn't contain all the information that's necessary to define our tetrahedral mesh. It's just for visualization purposes. We can also download our filtered consensus volume and solvent mask if those are useful for just checking our mask to see that it's not cutting off any parts. I'll go ahead and download this PDB file and open that up in a volume viewer. Along with this PDB file, I'll also go ahead and download the consensus volume density map and open that up as well. I've gone ahead and opened those in Chimera here. And what we can see is a depiction of our consensus density map. And this is a density where the flexible arm that's on the top right here is quite poorly modeled, or poorly reconstructed rather. And we can also see our tetrahedral mesh overlaid on top. And this is in the format of a PDB file where we just have used atoms and bonds to depict the mesh. Of course, it's not actually made of atoms and bonds, but just to depict the mesh. So we can see how the mesh is covering the object. And this is the mesh under which 3D Flex will model deformation of our protein. So 3D Flex will be able to move around the mesh nodes as well as deform them by moving around their vertices, and through that deform the density inside of each cell, and that's what yields deformed versions of our base density that 3D Flex will also be learning, the canonical density map, which will overall together be able to model conformational heterogeneity in this dataset. Now that the 3D Flex mesh prep job is finished, it's outputted everything we need in order to start training a 3D Flex model. Primarily, that's the 3D Flex mesh object, and that actually contains all the information about the solvent mask, the mesh definitions, rigidity, weights, and everything else. So we can go ahead and start creating a 3D Flex training job. The 3D Flex training job has more parameters than the other jobs and has some choices that we need to make. In order to construct a training job, we need to provide our 3D Flex mesh object as an input, as well as the particles from our data prep job. If we look closely, the particles, when they're connected, will use the blob_train and blob_train CTF, and everything else about the particles, including the full resolution blobs and other data, will be passed through. The CTFs and alignments, of course, are also used during training time. Once we've connected those two inputs, we can go ahead and set some of the parameters. So I'm also going to open up a training job that I've already run here that shows us what the output looks like and what some of the parameters were. So the first parameter that we need to set is the number of latent dimensions in the FlexRefine model. And you can see more details in the tutorial in the guide, but and as well as the preprint. The number of latent dimensions controls effectively the capacity of the model in terms of the number of different kinds of motion or sort of independent, simultaneously occurring, maybe independent is not quite the right word, but orthogonal, no, orthogonal is not quite the right word either. Sorry. So sorry. Now that the 3D Flex mesh prep job is complete, it's produced everything that we need in order to start the next phase of our process, which is training a 3D Flex model. Primarily, that's the 3D Flex mesh object that actually contains all the information about the solvent mask, the mesh itself, the rigidity, weights, and everything else that we discussed. So we can go ahead and start creating a 3D Flex training job by creating a job from the builder. And to that, we can connect our 3D Flex mesh as well as the particles from our data preparation job. And if we look closely, this will connect the blob train and blob CTF particles for training size, but not the full resolution particles. Those will be kept as pass-through outputs for use later on. in our job builder, we can also start setting the parameters of this training job. The first parameter, and the most important, is the number of latent dimensions. This is discussed more in the preprint and the tutorial in the guide. The number of latent dimensions controls effectively the capacity of the model by choosing the dimensionality of the ways in which the model can represent conformations. So when there are fewer latent dimensions, there are fewer different directions in which the model can change the structure at the same time. For example, with 2 latent dimensions, the model essentially can move around the latent coordinate in a 2-dimensional landscape, and every point in that landscape corresponds to a different motion or different conformation of the particle. But as we add latent dimensions, that adds more capacity to the model to simultaneously model multiple different motions that are occurring at the same time. Of course, as the number of latent dimensions grows, the cost of training the model also grows, but more importantly, the capacity of the model grows, and therefore, if there is too much noise in our data or not enough signal to support a more complicated model, as we add latent dimensions, there's also potential for overfitting or making a model that's learning implausible or unreal deformations. And this will manifest as worse FSC resolution and validation, as well as clearly incorrect trajectories and structures. So the number of latent dimensions, we recommend starting with 2 and then repeating training with more latent dimensions if you feel like the data appears to have more modes of motion present, or sufficient signal to allow more types of motion to be simultaneously resolved. The next parameter is the number of hidden units, and this is not the most important parameter, although you can reduce this if you feel like you're working with a protein that has less data, a smaller particle, less signal per particle. This also effectively reduces the capacity, the nonlinear capacity of the model to model deformations, and can help to constrain the model to only model simpler motions in the case where you have a simpler protein or simple motions that you expect. We don't tend to play with this too much, but there's an example in the paper of using a smaller number of hidden units and still getting an effectively good result. The next 4 parameters are all learning rates, and I'll come back to talk about what these mean and why they matter. We spent a lot of time tweaking the technique so that these don't have to be changed much, or if ever. But in case you do need to change them, these can be changed, and I'll describe in what scenarios that might need to happen. The rigidity or lambda parameter is one of the most important parameters to tune, and this is, again, shows up in the preprint and the tutorial online, is a parameter that controls the overall strength of the prior that controls rigidity within the 3D Flex model. This rigidity prior tries to enforce, along with the rigidity weights, which are relative weightings, it tries to enforce that tetrahedral elements don't have overly non-rigid deformations. And the rigidity weights help to indicate to the model which parts of the density should be allowed to expand and contract in a non-rigid way, or should be allowed to shear, etc., versus parts that have strong density and are already well resolved that should really stay relatively rigid. And so this is not a hard and fast rule. Of course, the model is trying to model non-rigid deformation, and so it has to have some flexibility in order to do that. But the penalty helps to stop the model from overfitting and control the complexity of the motions that it will model, and stops it from just overfitting to noise in the particle images. So the rigidity strength is very important. We have also spent quite a while getting the technique to work across different data sets within a reasonable range of rigidity values. So you might find that values between maybe 0.5 or 0.1 all the way up to 10 or 20 are reasonable values. But outside of that range, probably for most data sets, will be unreasonable. We start with a value of 2, and that generally works, but typically what you'll have to do is train a model once with a certain rigidity value, and this will also depend on the size of the mesh, topology of the mesh, etc., and the rigidity weights that you use, and then retrain with different values of the rigidity strength and see what kind of results you get. At some point, if the rigidity is too high, you will see that the model is barely learning any motion, or it's learning only very rigid motions. If it's too low, you'll see the model learning all kinds of intricate and interesting motions, but FSC resolution, when you look at local resolution improvements in the reconstruction under 3D Flex, will be poor because these motions are not actually present in the data. And so although the model tries to learn them from the lower resolution training data, those motions do not create high resolution correlation between the two separate half maps at high resolution, and that's a way you can tell that the motion is definitely not real. So, of course, validation of models like 3D Flex and other heterogeneity models in general is a very nascent area of research, and something that we're hoping ourselves and others will continue to develop techniques for. But this is a parameter where you will have to empirically try things to see what happens. There's also a parameter number of extra epochs that controls an additional extra time of training. By default, 16 epochs are used through a schedule of training that also includes frequency marching. So at the beginning, the 3D Flex model is limited to an even lower resolution than the training Nyquist resolution. And slowly over those 16 epochs, it's allowed to use more and more information up to 80% of the training box size. But you can add extra epochs as well in case you want the model to continue training after that. Of course, you can always take a checkpoint from a training job from a previous or intermediate iteration and use that for further processing. So it's fine to have the training job run for longer and use an earlier solution or earlier checkpoint if you feel that over time the results are getting worse after a certain point. And that is something that happens with some machine learning models in general. The next parameter, noise injection standard deviation, is a little bit of a nuanced parameter, and it's described in more detail in the preprint. It's part of our autodecoder latent inference technique. And the idea of this variable or this parameter is to control how much noise is injected into the latent variable estimation process. The purpose of the noise injection is to help the 3D Flex model during training retain some element of smoothness of the latent landscape. This means that positions in the latent landscape nearby to each other within this sort of standard deviation of, let's say, 0.15, those positions within the latent landscape should correspond to similar conformations. So if you can adjust this parameter, making it smaller will cause the 3D Flex model to have more precision in its estimation of latent coordinates. Not necessarily more accuracy, but more precision. And this can allow it to learn more interesting structure in the latent space, so like clustering and things like that, and sort of tighten up those features that it might learn. But it also means that if there isn't really enough signal in the data to support estimating the latent position for every particle with so much precision, then this can, again, add more capacity to the model than it should have and allow it to overfit or do other strange things. So we generally don't play with this value, but if you want, you can reduce it, especially for larger proteins with a more interesting conformational landscape, but also more signal per particle, you can reduce this value. And for smaller proteins with less signal per particle, you can increase this value. Finally, the last parameter that is typically necessary to tune is the latent centering strength. And this is, for lack of a better term, a sort of annoying parameter. It doesn't quite affect the results. What this parameter does is it controls a prior that's also part of the model that helps to keep latent coordinates estimated for all the particles centered around 0 in the latent space. And this makes sure the latent variables during training don't start to diverge very far away from 0 and become very large values, since that really is just a not useful sort of distribution of latent coordinates. During latent estimation, we only search for latent coordinates within the range minus 1.5 to 1.5. And so it's important that the model is constrained to only use that sort of volume in the latent space. So this latent centering prior has a strength that needs to be tuned. Unfortunately, this is a parameter that needs to be tuned on every dataset. Typically, larger values are necessary for larger particles or particles with more signal-to-noise ratio. We haven't yet figured out a way to automatically tune this parameter, but at the end of the day, if you're seeing that latent coordinates are too close, that are all being estimated very close to 0, meaning that the latent coordinates are all being forced to be too close to 0 during training, that means this parameter is too large. If you're seeing that many latent coordinates are going far away from 0 and hitting the minus 1.5 to 1.5 range of estimation, the endpoints of that range, that means this prior needs to be increased. I'll describe this again in a moment when we see the plots, but it's a parameter that, once tuned, can be left alone and really doesn't affect the results at all. But if it's not correctly tuned, it can just create problems in training. Right, so these are all of the parameters. One of the things that 3D Flex training allows is to input particles that already have an initial guess of latent coordinates or components. A typical source for these coordinates might be from 3D variability analysis. And in fact, one of the examples in the preprint shows how to use 3D variability, or rather that we did use 3D variability analysis initialization of latent coordinates to help seed 3D Flex in a more difficult case. So in some cases you can do that, and if you've already run 3D variability, you can simply connect particles to your data prep job that already have components mode 0, 1, 2, however many components you have, and then those particles, those outputs will pass through and arrive here, or you can connect them directly at this stage. And if you do want to use those latent variables, you can turn on the parameter initialize latents from input, and then you can also set a list, a comma-separated list of indices of input latents to use. So for example, if you connect particles with 5 initial values of components, like components mode 0, 1, 2, 3, 4, then you can choose to only use some of those to initialize the latent dimensions. For example, if you're only using 2 latent dimensions in 3D Flex, you can choose to use indices 3 and 4 from your initial input to initialize the 2 latent dimensions that you have for 3D Flex. So that's an option that's present. Once you've filled in all of these parameters, and these defaults do work well for this data set, so I can go ahead and launch this job, you can start the training job. And the training job is GPU accelerated. It uses only 1 GPU right now. And when it starts, it will output its logs to the stream log, and we can watch what it does. And I'll just describe that as it's going. So first, the model will load up our mesh that was generated previously and just display an image that helps to sanity check that the mesh looks fine. And then it will read all the particle data files and the CTF data files, and then initialize all the particle data. And then it'll start doing a test reconstruction. And this is a test reconstruction that is made basically to confirm that all the particles, alignments, poses, solvent mask, and tetramesh and everything are all correct. Let me just navigate there again. So let's check point 1. So in that test reconstruction, what you'll see is first a plot of iterations that only goes up to 80, and a loss. And what's happening during this test reconstruction is that we're using the same optimization and everything as 3D Flex, but just optimizing a canonical density, or a consensus density in this case, with all the particle data without any deformation or latent variables at all. And so this is just a test of 3D reconstruction using the 3D Flex GPU code and etc. and PyTorch and all the other dependencies. And if there's any problem, it'll show up here rather than during training time. What you should see during the test reconstruction is that the loss decreases. The black line is the training curve, the red line is a validation curve on a held out set of particles. And you should see that the density after training looks quite reasonable, and like a not so clean version of our consensus reconstruction from earlier. And as long as you're seeing that, then everything is good. Once that initial density is trained, you can compare it against the canonical density that was input. Then 3D Flex starts actually performing training iterations. And we can follow the latest checkpoint to just see the training iterations. So the first thing you'll see after the training iterations start is a line that continuously updates that tells you what iteration we're at out of how many, along with an estimated time of processing and the average speed of iterations and the speed of current iterations. Then you'll see a series of plots. And these plots at the beginning of training are updated every 20 iterations, and then afterward updated every 100 iterations. And these plots show a variety of diagnostics that are useful to understand whether training is progressing well or exactly what is going on. So now that this job has reached 100 iterations, I'll be able to explain some of the curves. And in fact, I will use a job that's already run to explain the curves so that there's some more data to look at. All right, so the first set of plots is really the most important. It's a pair of plots over iteration count. The first one plots training and validation loss. Validation here doesn't mean Fourier shell validation or Fourier shell correlation validation or any other kind of cryo-EM related validation, but simply the score or loss of the or objective function value of the neural network model evaluated on a held out set of particles from the input set. These particles are just used continuously to evaluate the model, and since those particles are not changing, the selection of particles is not changing, that lets us see a continuous line to tell what the model is doing. The gray line in the background is the training loss, and that has a lot of variance since from iteration to iteration we're seeing a different random mini batch of particles from the training set, and so we can't really tell what's going on by looking at the training curve, but the validation curves tell a different story. There are 3 different validation curves that are present in the plot, and these are all important to understand their meaning. First of all, the light blue validation curve is the full validation loss. And this means the total loss, including the data term and the rigidity penalty. Since our loss or objective function for 3D Flex training includes how well our model is matching the experimental data, it also includes a component of how much penalty we're paying for having non-rigid deformations. The rigidity penalty is contributing a part to our objective function. So the light blue line is the sum of those parts. The red line is the same as the light blue line, except it only shows the data term without the penalty. It only shows what does the flex model, how well does the flex model compare against the data. So when we see the red line decreasing, that means that our model is learning to match the data better. And the blue line similarly is decreasing. That means our model is simultaneously matching the data better, but also optimizing the rigidity penalty. The green line is a little more nuanced to understand. The green line is a baseline curve that helps us to get a sense of how much improvement the model is really making. The green line is the validation loss with everything the same as the blue line, but without any deformation at all. So this is just a rigid baseline. This is comparing how well does the canonical density that we've modeled so far without any deformation match the particle images. And this is a useful baseline because during training, many things are changing, and primarily the resolution at which our canonical density is being resolved, or the resolution at which it's allowed to be resolved, since we're using a frequency marching scheme, is changing. So over time, we naturally expect that the model is going to learn to match the data better for no other reason than that the canonical density in the rigid parts is going to improve. And of course, that doesn't tell us anything about whether or not the model is actually improving in modeling the flexible parts. And so the green line serves as a baseline to tell us how well is the canonical density doing in just modeling the rigid parts of the structure. And the distance between the green line and the blue or red lines is actually what matters most to us during training. And that's what's presented in the plot on the right. So the black line on the right is the validation loss of the flexible model, so that's the red line, minus the rigid baseline, that's the green line. So as that black line shows, the model is continuously improving in the sense that while the canonical density is improving on its own just to model the rigid parts of the structure, the flexible part of the model that's deforming the flexible regions is also improving relative to the rigid baseline. And that's really what we want to see, and that's how we know that the model, the deformation part of the model, is doing something useful. The second curve on the right-hand plot, that's the blue curve, is showing us the value during training of the rigidity penalty, and that's plotted on a separate axis on the right. So that blue curve on the right-hand plot is the same as the distance between the red and light blue curves on the left-hand plot. It's telling us how much are we paying in terms of penalty because our deformation is non-rigid. And this is also a very important thing to understand during training, or to watch during training, since at the end of the day, the model is optimizing both of these, the combination of both of these objective functions. It's trying to minimize the discrepancy between the model and the experimental data, that's the black line. It's also trying to avoid paying too much non-rigidity cost, that's the penalty. So we can see that in this case, the model is learning to match the data better, and it's also not increasing its rigidity penalty too much over time. It's balancing those well. We can also see by the raw numbers that in terms of objective function, and these are somewhat arbitrary units, although they're sort of related to a log probability score, but we'll just consider them to be arbitrary units of optimization right now. The model is doing better by about 20 units over time as it trains versus the data, and it's paying a cost of about 1 unit for the penalty. So this gives us a sense in terms of when it comes to the model's optimization, it gives us a sense of how much the model is paying attention to the rigidity penalty versus the data. And this is what we can look at to see whether or not the strength of the rigidity penalty needs to be tuned. If we're seeing that the model is not optimizing the data curve and is paying too much attention to the penalty because these values are too high, then we can reduce the strength of the penalty, and vice versa. Some of these effects are quite nuanced, and at the end of the day, all of this has to be done empirically, and you may have to try training 3D Flex multiple times with different settings and see what the results actually look like visually, more so than these curves, to really tell what the difference is between different parameter settings. But these curves do help in practice as well. Now I'll switch to the Flex training job that we had running previously to look at its diagnostics plots as it's been training over a few thousand iterations that have gone on so far, and see what we can tell from the diagnostic plots. So here again, we see that our training curves and validation loss curves are doing well. We're seeing that over time the model is slowly learning something above the baseline, the green baseline, which shows in this black curve here. And we can look at the rest of the diagnostic plots as well. So first of all, there's a plot of the current canonical density, and that's the density that the 3D Flex model is learning from the data, albeit at a lower resolution. This is at the training resolution, so we won't see high resolution detail there. But we may see some flexible parts like this flexible arm or leg region of the integrin starting to form structures that are not present in the consensus density. There are also some diagnostic plots about the deformation model itself. The first plot here shows the values that come out of the generator, the flow generator, in its last hidden layer before the final output layer. And this is a 64-dimensional output hidden layer, given the parameters that we set during training time. What this plot shows is the activation of those hidden units as we vary the latent coordinate across dimension 0. And this just serves as a quick check to see how nonlinear are the outputs of our deformation generator network. What we should see here is that during training, at the beginning, these lines are mostly straight lines, as the network is just learning simple linear patterns, but over training time, we start to see it learning more nonlinear deformations, and those are represented by the curved lines. This, of course, is not a complete picture of what the deformation model is learning, since it does have more than one latent coordinate and more than one hidden layer. These are the last hidden layer activations. So they're summarizing what the network is doing across the latent space, but as it moves in other dimensions, for example, in this case we have 2 dimensions, or in a combination of latent coordinates 0 and 1, other nonlinear changes may also happen, and those are not captured in this plot, but it just serves as a quick sanity check to see that the network is learning something. On the right-hand side, we have another diagnostic that just shows with 3 images of our protein molecule, or the tetramesh rather, the deformation that's being learned in different parts of the structure. So here, as long as we're seeing some values, and the color bar shows in units of pixels the amount of deformation being learned in different regions, we can see that here in this block, the deformation model is learning to move this flexible arm, and that's what we expect to see. So it just gives us a sense that what's the magnitude of deformation essentially that the model is learning. The next few plots are to do with the latent distribution. So the first few of them, and there will be k of them for a k-dimensional model, are just one-dimensional histograms along each latent dimension to show us what the latent variables are looking like. And then the next few plots will be plotting latent dimensions against each other. So 0 versus 1, 1 versus 2, etc., similar to how the plots are in 3D variability analysis. What we see here is related to the parameter that we discussed earlier, which is the centering strength, or in the latent space. And that's a parameter that, as I mentioned, is a bit of a nuisance, but it needs to be tuned on a per dataset basis. And here we can see that it's tuned well because our latent variables are not too tightly centered around 0. They range in value from about -1 to +1, and they're not hitting the edges of our -1.5 to 1.5 range of latent estimation. So these latent distributions are good, and beyond that, the centering strength does not really matter, since the 3D Flex model has complete control, or rather, it is able to learn any pattern in the latent space and then map that into any pattern of deformation. So if we squish or stretch the latent space a little bit, as long as the flow generator also learns a corresponding change, the deformation model overall can model the exact same motions. So as long as we're not hitting the edges of the range or being too closely packed around 0, everything is good in this latent space. So these are all the training time plots that help us understand what's going on during training. And essentially, while this training is happening, we can either watch these plots or just take a break and come back, or we can start using some of the visualization tools that allow us to look at what the model is learning while it's still training. So the next job in the set that we'll discuss is the 3D Flex Generate job, or generator job. This is a job that we can build, again, from the job builder, that takes in a 3D Flex model, and that's the output of 3D Flex training. This particular generator job has a parameter called override scheduler, and that's on by default. And when this is on, as soon as you queue this job, it'll start to run right away, even though the training is not finished. And that will mean that it uses the current checkpoint that's been output by the training job. So in this case, our training job has done some 6,700 iterations at this point. And if we look at the outputs, our 3D Flex model has many checkpoints, one checkpoint every 100 iterations. So all of these checkpoints are available, and we could actually choose to use an intermediate checkpoint as the input to a generator job if we'd like to see a non-final result. But if we just connect them as is, then the generator will use the most up-to-date checkpoint that it can at the time that you queue the job. So along with the 3D Flex model, the generator job also takes in an optional high-res or full-res volume and latent coordinates. I'll describe those in a moment. But by default, the generator just has a couple of parameters, or just one parameter really, which is the number of frames to make per volume series. And with nothing else input, the generator job will simply create volume series that traverse the latent landscape along each dimension. This is very similar to how 3D variability display works. So I'm just looking at a completed 3D Flex generator job right now. What the job will do is first load up a checkpoint from our training model and display some plots just as a sanity check, the tetramesh that's been loaded, as well as all of the training plots that we previously discussed for that checkpoint, just as a reference so you can remember what was happening at that checkpoint, all the diagnostics and so on. And it'll also output a plot showing the series of volumes that it's going to generate. So by default, it'll generate, like I said, one series per dimension. The first series will be across dimension 0. So those are the orange dots, and the next one will be green dots along dimension 1 in the latent space. And if we had more than 2 dimensions, there would be correspondingly more series. And then it'll go ahead and generate, meaning it'll run through the 3D Flex model using the canonical density that's part of the model, or the currently trained canonical density. Map that through the deformation generator at these different points in the latent space, and output the convected 3D density volume as a series. And those series are zipped for space. They don't take too much space in zipped format. but they do expand to a very large size just because of the number of volumes. By default, the job will create 41 volumes. We like to use an odd number here so that at least one of the samples will be at the origin in the latent space, just so you can see what the undeformed or canonical version of the structure is. And again, I already described what the override scheduler option does. So this job that I'm displaying here is already run, but I can go ahead and queue up this job that I've created, and that's against our currently running job number 156, 3D Flex training. So you can see that this Flex Generate went ahead and launched, and it'll start producing those generated volumes. Then we can take a look at what the outputs look like. I'm going to open up job number 131, which is a previously run Flex Generate job from a previously run training on the same dataset. We can see here again that it went through the same steps and generated volume series. You can go ahead and download those volume series, and those will download as zip files containing all the volumes in the series. I've gone ahead and done that and opened those up, unzipped them and opened them up in Chimera again using the volume series tool, which is very handy. Here it's often useful to set it in oscillate mode, cache many renderings, more than the number of volumes you intend to load, and increase the data cache size enough to hold all of those renderings. I've loaded up both volume series here, so we'll start playing the first one. This is volume series along dimension 0, and the frames range from 0 to 40, which is a total of 41 frames. So here we can see the type of nonlinear, non-rigid deformation that the model has learned for this particle. And we can see that it's learned motion of multiple different parts at the same time. And this is just one dimension of motion and just one straight line trajectory through the latent landscape. And of course, across the latent landscape and in different directions, the deformation may be different. There may be other kinds of motion present, and we really have to explore that latent distribution using this generate job to see exactly what the model has learned. So that's latent dimension number 0. We can switch over to dimension 1, play that series. You can see that that's a different type of motion that has been learned here. And both of those motions, of course, and the combinations of those, which may not be simply the sum of those motions, are present in the data at the same time. And every particle, when we're estimating its latent position or latent coordinates, it's finding a position along these deformations for the particle in that particular image. Now that we've seen the output of the generate job, I'll just quickly describe a slightly more advanced use case for the generate job. I will clone this job, and we can look at the job builder for this job number 158. We can see that there are two optional inputs, a full resolution volume, and we'll come back to this once we've learned how to use the flex reconstruction job, as well as latent coordinates. And these are inputs that you can provide by creating a custom trajectory through the latent landscape to enable the generator job to generate volumes along a volume series that isn't just a straight line in the latent landscape. So for example, this whole workflow of creating custom latent variables is described in the CryoSPARC tools documentation. And there's actually an example Jupyter notebook that runs through how to use CryoSPARC tools to plot out the latent landscape from a 3D Flex training job, and then interactively create a trajectory. And here I've just created an arbitrary trajectory. You can do whatever you'd like to create a trajectory through the latent space, or to explore any part of the latent space that you want to see what the volumes look like in that generated area. And then the example notebook continues and explains how to inject that result back into CryoSPARC. And you can use that output from the external job that's created through this notebook and connect that as an input to the latent coordinates of the generator job. That'll cause the generator to, instead of creating a volume series along just these straight lines of dots along the latent trajectory, to instead use the specific trajectory that you choose. And you can use any method that you'd like to generate the trajectory. The final job in the series that we'll talk about is 3D Flex reconstruction. 3D Flex reconstruction, as mentioned, is a job that's responsible for computing a high-resolution reconstruction in 2 half maps under the 3D Flex model using the full resolution particles from data preparation. So in order to construct a reconstruction job, again, we can use the job builder, create a 3D Flex reconstruction job, and this time we again need to connect a model from our training. So we connect our 3D Flex model, and this can again be a training that's still running in case we want to run this. This job has the same override schedule parameter as the generator job, though it's off by default. And we also need to connect our prepared particles. We can actually connect our particles directly from training, and we'll see here that this time it's the full resolution blobs and CTFs that are connected rather than the training time downsampled versions of the images. The reconstruction job really only has two parameters, or three parameters. One is whether or not you also want the reconstruction job to do a rigid reconstruction of your particles just as a baseline. This is often helpful to be able to get a direct comparison between rigid and flexible refinement results. The number of BFGS iterations, generally we don't find that more than 20 are useful, but perhaps on some larger proteins or larger box sizes, more iterations can be helpful. This does come with a computational cost, but it's generally not that large, so you could increase this to 30 or 40 in a final result, for example. Finally, there's an option to force redoing the gold standard split. By default, particles are split in half into the two half maps using the same gold standard split that was used in the upstream refinement, since the particles actually do contain their alignments which note which half map they were part of previously. And so we don't mix the particles unless you choose to force redoing the gold standard split. And this is necessary if for some reason the particles you've inputted don't have an equal number of particles in split A and B in the input. So you can choose those parameters and then run the job. And I'll just show what that job looks like after completing. It again loads the mesh as well as the model from training time. It then reads in all the particle data, and this can take some time since all the high resolution particles are now being read into CPU memory. And in fact, this is an important note that's also in the tutorial and the guide. 3D Flex training and 3D Flex reconstruction both load all particles into CPU memory at the same time. They don't read particles from the CryoSPARC cache yet. And so these jobs do require a lot of CPU RAM to hold your entire data set while they're processing. This makes them faster because the particles are just immediately available to process, but large data sets require lots of RAM. So here we can see that there were 42,000 particles in each half set, and BFGS optimization went through 20 iterations on either half set and computed these two half maps, one for the A side and one for the B side. These two half maps are output from the job directly, as is an FSC curve, which is computed between these two half maps using the soft mask that was defined in the mesh preparation job earlier. And this may not be the best mask to use for FSC calculation, so you can create another mask and use the standard FSC validation job from the job builder in order to compute any other FSC. You can also use these two half maps for any downstream operations like local filtering, local resolution estimation, sharpening. You can even use the new Deep Enhancer wrapper for sharpening if you'd like. Any of these pre-processing tools can be used anytime. Finally, the job also outputs a filtered full map. That's a combination of these two half maps filtered by the FSC resolution. And then once it's done that flexible refinement, it'll also commence the rigid refinement, which uses the same BFGS optimization, but just uses the particles without any deformation model. And again, computes an FSC resolution and a single map. Here you can see the difference if I just quickly open these two FSC curves, the first being the rigid reconstruction and the second being the flexible reconstruction. You can see there's an improvement in the FSC overall for the overall protein, and that's because multiple parts of the protein are being improved by FlexRefine. And you can also see in the density maps, if I look at the filtered full map for both cases, this time the flexible refinement is first, and this time in this case the rigid refinement, you can see that parts of the structure are substantially improved by FlexRefine. Right, so that's the 3D Flex reconstruction job. Its outputs, as I mentioned, are maps and half maps for both the flexible and rigid case that you can use for any downstream processing. You can also take the volume straight out of reconstruction, and this will be a high resolution volume. You can take it directly, or you can sharpen it first, or whatever you'd like, and then create a 3D Flex generator job. This time, connect the full resolution flex volume to the volume input, and connect the model from your training job to the model input. And now, when you run the generator job, this job will, instead of using the canonical density at low resolution that was part of the trained model, it will use the high resolution reconstructed density from the flex reconstruction job and deform it by the same deformation model that was learned during training time. The deformation model is upscaled to the new box size of the high resolution density, but this job will allow you to visualize the same bending and deformation motions, this time with a high-res model. This is great for making nice videos and perhaps for publication figures or anything like that. I've run this job now, and it'll run and generate some outputs for us, which I can then download and we can take a look at. I've gone ahead and downloaded these two volume series, and we can take a look at them. What we're seeing now is the high resolution reconstruction from 3D Flex Refine, where the flexible parts of the molecule are improved. We can see that although this is an unsharpened reconstruction, it's just a raw map that's a combination of the two half maps filtered by the FSC resolution. There's plenty of detail in the core regions and the flexible regions as well. It's not sharpened, but we can take a look at the motion. If we go ahead and play this series, we can see that we're seeing now the same motion that we saw earlier at a low resolution, but this time seeing the high resolution version of the density deforming along that trajectory in the latent space. So that's the 0th component of motion. And this is the first component. Obviously an interesting result, and what we'd like to see out of a tool like 3D Flex. That concludes all the different job types that we wanted to cover in this tutorial, explaining how to use 3D Flex. Please stay tuned for the second tutorial that covers how to use segmentation to create custom meshes and custom topologies for meshes. And enjoy your processing. Thank you.